Good afternoon, everyone. My name is Ying, and I'm an assistant professor at the Department of Biostatistics and also for Center for Computational Molecular Biology at Brown University. I will try my best to finish my tutorial on statistical and computational methods for cell type deconvolution in spatial transcriptomics. So here is the outline. I will first introduce about what is the spatial transcriptomics and how can we get the data from different platforms, and also talk about the problem formulation, exactly what is the cell type deconvolution and why does it matter, and also I will give you an overview of the method based on different categories, including one of the methods we developed before, and also about these evaluation and benchmarking, and also at least some points of my personal perspective about these open problem and future directions for the field. So spatial organization of cells is very important for the multicellular life because it affects the function and also the evolution. And recent advances in spatial transcriptomics technologies enable us to measure the gene expression while also preserving the spatial information. So after we get these kind of data, you will know, for example, the spatial location for each cell or a bunch of the cells. So there are so many technologies. Briefly, they can be categorized into two categories. First one is this sequencing-based technology, and second one is this imaging-based technology. So the sequencing-based technology is directly based on quantifying these transcripts and also combined with the spatial barcoding to get these spatial information, but it might also sacrifice this spatial resolution. However, for the imaging-based technology, it directly utilizes the in situ hybridization or in situ sequencing that directly detect these transcripts at each spatial location. So it gives you the high spatial resolution of this data. So these two technologies have their own characteristics. So they are different platforms. So let's look at these. If we have this tissue sample, and let's say if we generate these data from this 10x Visium technology. So finally you will get these, each spatial location, we call it a spot. And you will see that because multiple cells that can be located into the same spot due to the low resolution. So finally, actually, for each spot, spatial location, it captures the mixture expression across a bunch of the cells. And depending on different platforms, the number of cells can vary. For example, for the 10x Visium technology, it goes from 1 to 10 cells. Or for higher technologies such as the SLICE version 2, it probably at most 3 to 5 cells. However, on the other hand, for the imaging-based technology, because it directly detects the transcripts through these imaging-based fluorescent in situ hybridization, so you will see that you can directly locate the transcript position at each cell. And after the image segmentation, you can finally get the single cell resolution gene expression data. So the main difference between these two technologies, the first one is this resolution. For each spatial location, sequencing-based technology might, for example, the 10x Visium, typically 1 to 10 cells. But for imaging-based technology, after image segmentation, you will finally get these single-cell resolution data. And also the transcriptome coverage is also different for the sequencing base because it's directly based on next generation sequencing, so it can give you the whole transcriptome coverage. But for this imaging-based technology, because it depends on the pre-designed gene props, so it can only give you targeted gene panels, hundreds to thousands of the genes. So corresponding to how we understand the cell type, different data types because of their different characteristics, the angle to get the cell type information might be different. For the sequencing-based technology, we can estimate the cell type proportion through the cell type deconvolution, which I will be defined later. And for the imaging-based technology, because after imaging segmentation, each location represents one cell, so we actually call it cell type annotation. So all these cell type annotation works that you can finally apply for this imaging-based technology, and you can annotate what each cell represents. So these are the timeline for these spatial transcriptomics technologies, corresponding to, also combined with their commercially available platforms. So starting in early spatial transcriptomics technology actually start from academic labs. And starting in 2016, later these science paper, the 10x Visium turned these platform into a platform. And all recently they are also high-resolution platforms. So this is a simple illustration of 10x Visium pipeline. Briefly, it enables the gene expression profiling on tissue locations with spatial localization information. And for example, we can get this microscopy slide from the company, and each slide containing about 6 different squares. And when we zoom in the square, it's containing about 5,000 barcoded props. And then you place the tissue on the square, the mRNAs released from the tissue will be hybridized with the barcoded props underneath the tissue. And each capture area represents a spatial location. And this spatial location, you can see that the diameter is 55 micrometers, which is much larger than a cell size. So that's why this technology can only give you the measurement across a bunch of cells instead of single cell resolution. And finally, you will get the gene expression data through these, in these 2 data matrices. So the first one is this gene expression data, which each represents a matrix that measure the gene expression across all the genes for each spatial location. We call it spot here. And correspondingly, because the capture area, you also know they are spatial coordinates in the 2D array, so you will also get these 2-dimensional spatial relationship. So that's the typical 2 data matrices we will get from any spatial transcriptomics technology. One is the gene expression data, another is these corresponding the spatial coordinates information. So let's look at the cell type deconvolution. Because, as I mentioned at the beginning, for the 10x Visium technology, because of the resolution limitations, each location is not a single cell resolution. Instead, it measures a mixture of the cell types. So in order to know what exact, like let's say we want to understand how the cancer cell distribution, we need to disentangle what are the cell types existing at each spatial location. So that's why we need the cell type deconvolution, which is trying to estimate the cell type composition within each spatial location. So given this two data sets, one is gene expression, another is coordinates, and given each spatial location, we measure a mixture of the cells. And through these deconvolution algorithms, we want to estimate exactly what percentage of each cell type belong to at each location. So that's how deconvolution task is defined. So before we develop methods for spatial transcriptomics deconvolution, there existing a technology, bulk RNA-seq. We all know that it measures, it's even before the single cell. So it measures the gene expression across a large number of cells, which is a mixture. And there are also methods that performing these cell type proportion estimation for bulk RNA-seq deconvolution. So why the previous solution doesn't work for spatial transcriptomics data? So in the traditional context, the deconvolution was originally developed for bulk. However, this bulk RNA-seq data represents a whole tissue sample, and there is no spatial information provided. And also, there are common assumptions for this bulk RNA-seq is that each sample is a mixture of the known cell types, and there are no spatial dependency among samples. And typically, the data is from a sample level data, no sublocation resolution. So this is the pipeline for the bulk RNA-seq deconvolution. Given the bulk mixtures from each individual, now the sample represents each individual or each subject. You want to estimate per each individual the cell type composition, not per each spatial location. And however, the differences between the bulk RNA-seq and spatial transcriptomics, let's say we have one sample from one patient, and we give the bulk, we do the bulk RNA sequencing. So finally, you will just get the gene expression for this one sample. It's just a vector of this gene expression. However, for spatial transcriptomics, the gene expression is a matrix that across all these spatial locations. And the cell type proportion we want to estimate is for each spatial location, not an average level across this sample. So the differences between bulk and single cell spatial transcriptomics is first, the sample size definition is different. In bulk RNA-seq, the sample is at each subject, each patient, each individual level. However, for the spatial data, the sample here represents each spatial location, and each spatial location you have their gene expression measurement. Also, compared to the bulk, the spatial transcriptomics has this low RNA content and high sparsity. So each location captures fewer cells, leading to noisier and sparser gene expression. And also the spatial environment. The nearby location might share more similar cell type compositions than the distant ones. And also, for the ballpark sake, previously there are typically lack of the histological integration about these molecular morphology. So all of these makes the previous solutions not work. So that's why people start to develop cell type deconvolution methods specifically for spatial transcriptomics, because the previous deconvolution method failed. And based on the different data type and data input and the output, we can categorize these three, current deconvolution method in three categories. So the first category is we call reference-based method. It utilizes external single-cell reference profiles to guide decomposition of spatial mixtures. So which means that given these gene expression data, if you have a prior knowledge about what cell types existing in the tissue and what are their cell type specific gene expression, that you can obtain that from the single cell profile, you can finally get these cell type proportion. So that's we call it this reference-based method. However, on the other hand, sometimes for some tissues, and especially for the understudied tissues, we might not have a gold standard single cell reference, or there might be a mismatch between the reference and the spatial data. Then then people are thinking about, can we just develop these cell type deconvolution method not to rely on these single cell reference? So we call these reference-free methods. They will infer the cell type composition without any external single cell reference. And the third category of methods are recently developed is because sometimes two cell types that they are very similar in their gene expression, you cannot just through these basic differentiate between their cell type expression to estimate their proportion correctly. So people are thinking about adding these histological information which can provide you more information about morphology and combine with other modalities to guide these cell type deconvolution. So these are a recent Nature Genetics review paper just published in May this year. They summarize these deconvolution methods, almost around 67 methods, and based on what information they use, they can be categorized like reference-based, reference-free, and also other information-based. That's just what I summarized in the previous slide. However, based on the modeling framework, the deconvolution task can be also categorized into different categories, such as the regression-based, and also the Bayesian framework, dimensional reduction methods, deep learning methods, also optimal transport-based method, and also the consensus method, which means such as ensemble learning to ensemble all the categories of the methods to get a consensus cell type proportion. So let's first briefly look at what's representative methods for these reference-based deconvolution methods. So the key idea is to leverage the existing single-cell reference data, which provides you the cell type specific gene expression profile for the deconvolution of these spatial transcriptomics data at each spatial location. So the first several methods, the first category is the regression-based. Here I just list one representative method called Spotlight. The key idea is use seeded non-negative matrix and factorization and also non-negative least squares into a three-stage regression pipeline. So the first stage is we perform a seeded NMF on single-cell RNA sequencing reference. So the goal is to extract gene by topic profiles. And the topic, you can connect this topic to like a cell type specific, cell type like concept. So they want to decompose, perform a dimensional reduction on the single cell data to infer these gene by topic profile. And the second stage is linking this topic profile with the spatial locations from the spatial transcriptomics data. So here is the spatial transcriptomics data with the predefined gene by topic specific expression. We can get this topic by spatial location composition. And then the third step is actually trying to mapping these topics to different cell types. So here we already got this topic by spatial location from step 2. And because for the single cell RNA sequencing, you also have these, you know what cells are from each cell type. And then based on this H file, you can aggregate a topic by cell type profile by just calculating the mean. So finally, you can get this Q matrix, which is the topic by cell type profiles. And then the next step is very simple. We just need to perform a non-negative least squares, get the regression weight. So finally, you will get these for each spatial location, the cell type composition. That's your final cell type proportion. So this is the results from the spotlight. So here, this is one data set from the 10x Visium from the mouse brain. And each location, you can see they have a small, very small pie plot. And basically, they can infer that, for example, the L2 layer cell type is located in L2 layer, and the cortex layer cell type, they are located very accurately in the cortex region. So, because the data set is actually the count data, there are also another category of methods which is based on the Poisson-based model. Another representative method is called RCTD, which is the robust cell type deconvolution for spatial transcriptomics. The goal is still deconvolve spatial transcriptomics data, also leveraging the single cell reference. And the assumption is each spatial location is a mixture of the known cell types from the reference. And here are some model notations. Given that we observed the gene expression data for gene J in spatial location I, which is Yij, and also given their rate parameter lambda ij, we assume a Poisson distribution, and we further place this log normal distribution on this rate parameter. Here, this is the model that link this rate parameter to this cell type proportion. So for each gene J on each spatial location I, we first link it to this spot-specific effect because there will be different detection sensitivity across the spatial locations. So this parameter accounts for these different detection sensitivities. And then we also assume that this part is explained by what the cell types contributed to the rate parameter. So the beta here is this cell type proportion in spatial location I and K. So that's our cell type proportion we want to estimate. And the mu Kij is representing the cell type specific expression that can be estimated from the single cell reference data. And the gamma J is representing this gene-specific platform effect because there might be, for example, different sequencing depths and also the batch effects between the single-cell RNA sequencing gene expression and also the spatial transcriptomics gene expression. So they also account these gene-specific platform effect. The key innovations for this method is they explicitly modeling this platform effect to account for the heterogeneity between single-cell reference data and the spatial transcriptomics data. And also they support singlet, doublet, and full mixtures through this model selection, which means that they can select different models by assuming how many cell types existing in different locations. So this is the RCTD results. In their main analysis, they're mainly focusing on deconvolving relatively higher resolution. So this is the slide SIG data from the hippocampus. You can see that they can locate these three CA3, CA1, and data gyro cell types very well. Also, they are confidently assigned of these spatial clusters to different interneuron subtypes. So, because this is the count data, there will always people also develop negative binomial-based model to account for the over-dispersion. So another method published in Nature Biotech called cell-to-location, they aims to estimate the absolute abundance of fine-grained cell types in spatial transcriptomics, also by integrating the single-cell reference data and spatial data. So instead of the Poisson model, they model these observed gene expression counts DSG for each spatial location S of each gene G as a negative binomial model. And then they further model this mu as another equation that consider this regression weight of this signature and also the reference cell type signature. So here, this WSF representing what cell type proportion for each spatial location S. And they also add another mg, which represents the technology sensitivity, because there are heterogeneity in terms of the detection sensitivity between single-cell platform and also the spatial transcriptomics. And also, all the other variances that's not explained by the cell types and also by the technological sensitivity, they aggregate, they have another term that's SEG to represent the additional shift. So they also assume the prior distribution of these cell type specific weights to decompose it into the location level on these latent factor. So the main prior assumption for this is trying to encourage the cell types that co-occur across these tissue regions to be modeled together. So if these two cell types that are occurred into the same tissue compartments, they should have a very similar cell type specific weights. And they finally use the variational Bayesian inference to do these, to estimate the cell type proportion. So this is the results that's published in their paper. So they analyzed a 10x Visium human lymph node data set, including these T cell and B cell zones. So let's look at their results that given these two annotations that represents the germinal center, and their method can locate these cell types very accurately into this germinal center. However, for the other methods such as the RCTD and the spotlight, you can see that the cell type proportion are actually outside these manually annotated region, which indicates that they are not less accurate. So another categories of the method is we developed before called CART, which is based on non-negative matrix factorization. So far we have introduced RCTD, spotlight, cell to location. However, all of these methods, actually their model doesn't take any spatial coordinates information. They don't explicitly model the spatial coordinates. And also, the listed deconvolution methods cannot give you a refined spatial map. What does that mean is they can only generate for each location a cell type proportion estimate. They still cannot give you a high resolution imputed gene expression that exactly representing a near single cell or subcellular resolution. Also, all the previous methods, they are require a single cell reference space. So all these limitations motivate us to develop this CART method. So first, I'd like to say spatial correlation does exist in the cell type composition. So we look at Murphy's data, which represents a single cell resolution, and we plot the cell type composition across n nearest neighbors. So you will see the proportion are much more similar in the nearest neighbors than the distant ones, which indicates that there might be some spatial decayness as the locations goes further. So that's motivated us to incorporate the spatial coordinates explicitly into our model. So our baseline model is still a non-negative matrix factorization model, but instead we also assume that for each cell type K, its ith location's proportion is dependent on these neighboring locations to borrow information from the neighboring locations. So we assume a spatial autoregressive modeling assumption on these cell type composition. So by combining these spatial information as well as the gene expression and also the single cell reference, our output is this cell type composition matrix. Also, as I mentioned that this can only give you the proportion at each location. But instead, actually, the location itself, between them, there are some gaps that cannot give you the gene expression measurement due to the technical limitations. So by utilizing the conditional relationship between these existing locations and our newly graded locations, we can actually impute the gene expression at a higher resolution. So the second goal of our method is that we can give you a high resolution of the cell type composition map as well as the gene expression. So I will, because of the time limit, I will just skip these methodological details and also the results. Maybe show these. So one of our important applications is in the pancreatic ductal carcinoma. And we found that our method is the only method that can identify these intratumor heterogeneity. And the upper region is mostly dominated by both cancer clone A cells and cancer clone B cells. And the bottom region is mainly dominated by the cancer clone B cells. And later, after our method was published, another method, they integrating these histology information, but not working on deconvolution. They also find that there is indeed these two region-based intratumor heterogeneity. So another task about this deconvolution is, can we do a high-resolution refinement? So here represents this low resolution, which means that each location represents a mixture of the cells. And the high resolution is that we want to know what exactly at each location, what is the expression for a single cell resolution, and what is the expression or near single cell or even subcellular resolution. So by doing this, we can impute the newly graded locations that not measured by the original technology to give you a high resolution map. So these are the results that clearly on the high resolution map, we show a more stronger pattern than this in these cancer clone B cells. And also we can see there is a clear boundary between these tumor core region and these tumor adjacent regions. So the next category is the reference-free deconvolution method, which means that there is no existing single cell reference. Can we just estimate the cell type proportion directly from the data without borrowing external reference information? So why we want to do reference-free deconvolution? Because there are some limitations of the reference-based deconvolution. First, they might depend on the accuracy of these single cell reference. They might require matching tissues, platforms, species, or conditions. Second, the biases can arise from such as the batch effects, low coverage, or missing rare cell types. For example, if your spatial data contains some rare cell types that was not measured by the single cell reference, then the deconvolution might have some bias. Also, the reference-based method assume all the cell types are known as a priori. They cannot capture novel, context-specific or perturbation-induced cell states. And also, there are some cross-modality limitations. The spatial and single-cell technologies differ in their sensitivity, gene coverage, and bias. So all these are motivate people develop reference-free deconvolution. They can directly infer the latent cell types and their spatial distributions from the spatial transcriptomics data, and it enables the unsupervised discovery of spatially distinct cellular communities, and also they are not requirement for these external single-cell references. So one of the representative methods is called ST-deconvolve. So the aim is to deconvolve these cell type transcriptional profiles and their proportions in each spatial location without using the single cell reference. So it is, this is the architecture. It is mainly based on this LDA model. So I will talk a little bit detail for in the next slide. So they adapt this latent Dirichlet allocation. We know it's a typical model that's assuming that each spatial locations are treated as a document, and the cell types are treated as topics, and the genes are treated as words. Then you want to see, given the documents that's representing your gene expression, what each topics that contributing and what the proportion of each topics that finally forming these documents. So let's say for each spatial location, the theta d is the cell type proportion that we want to estimate, and we assume that it follows a Dirichlet distribution, which guarantee that the proportion sums to 1, and also a typical assumption for the cell type proportion parameters, variables. And then for each molecular in these in the pixel D, we are assuming that the ZDM represents the latent cell type assignment or the topic for the M's molecular in the spatial location D. And then it follows a multinomial distribution with this proportion that you want to infer. And then given these latent assignment and also given the gene distribution, for example, the expression profile, you can assign these counts finally through this multinomial distribution. So this is your WDM, that's your finally assignment, the count. And the beta K represents the probability distribution over all the genes given the cell type proportion, cell type K. And they use this variational EM algorithm to infer the cell type proportion. So the key idea is to utilize this LDA model, treating the gene expression as a document and the cell types as topics and the genes as the words, and finally get this proportion for these topics. So this is the results for the ST deconvol. The main selling point for this method is they want to say that when you have a good quality reference, your performance is actually comparable with all these reference-based methods. However, if there are, let's say, missing cell types, or if you are even using not a good quality cell types, then they want to say that they can achieve a better accuracy in terms of the deconvolution. So there are also limitations for the reference-free method. First, it purely relies on the unsupervised latent cell types. Actually, what the cell types you infer can only be like topic 1, topic 2, which are unnoted. We still don't know whether which cell type, whether topic 1 represents cancer clone 1 or topic 2 represents cancer clone 2. So they often require post-hoc matching to known biological cell types using gene markers. So you still need to rely on some prior knowledge to annotate the topics and match them to the cell types. And also there are some interpretability challenges. It's difficult to validate or annotate without ground truth. And also the inferred profiles may mix multiple similar cell states. Let's say if your topic 1 and topic 2 are very similar, it's very challenging to annotate to the correct cell types. And there are no guidance from the prior knowledge because it doesn't leverage any existing knowledge. And it also very sensitive to the parameters such as how the number of topics you choose. It's still an open problem in the dimension reduction field, like how many topics you should choose. And it also might be sensitive to the gene selection because now you don't have this reference information. All your information very rely on what informative genes that can representative of these cell types. So that's why it's also sensitive. And it will also risk of underfeeding maybe too few components or overfeeding, there are too many components. So that's the limitations of the reference-free method. So I think 2 years ago, there are some methods published in very good journals like Nature and Biotech. They starting to add histological information for the deconvolution because the mRNA alone may not fully capture the functional cellular identity. And the spatial transcriptomics data is often noisy and sparse, especially for these lowly expressed genes. And the cell types with similar gene profiles may be harder to distinguish without additional information. So the histological features such as the morphology, tissue boundaries can be leveraged in the deconvolution. So for the 10x Visium technology, they will also have some paired histology H&E imaging data. And you will also have the paired spatial transcriptomics data. So there was a method called Starfish. The key idea is to develop this deep generative model integrating these transcriptomics data and paired histology information to infer a joint latent space. So here, let's say this ZI represents spot I's latent factor, and this latent factor is actually determined by their cell type state. And then, given the same latent factor, they can generate these H&E images, which is represented by the morphology information. And at the same time, you will also, given this latent factor, generate these gene expression information. So through this deep generating model, they actually integrate these image information and gene expression to learn a joint latent space. So this is the results from their paper. So you can see from the histology, especially for the tumor data, it's very clear that they can provide very good information about the boundary of these, for example, tumor core region and the tumor adjacent region. And correspondingly, this is their results, which you can see that they are very much matched with the histology information. So another method, which is published in 2024 called STY, which is called single cell level deconvolution. So the uniqueness of this method is that all the previous methods, they can only give you a roughly proportion estimate at each spatial location. But cell type proportion doesn't equal to the single cell level deconvolution. So here, what does it mean is that they believe that each location has multiple single cells. And these single cells, you can only get the information from imaging segmentation. So their novelty is they first perform the imaging segmentation identify these cell masks and the nuclei information, and then also get these imaging-related embeddings, such as representing the area or size or the axis. And then they do two deconvolution. One is based on gene expression profile, and one is based on the imaging profile. And then they minimize the similarity between these two profiles. Because you have the imaging segmentation information, you actually know what single cell existing in the spatial location. So that's why, finally, in their results, they actually estimate the individual single cell information in each location, and then the proportion was calculated based on that. So let's summarize these the two differences between cell type proportion and the single cell level task. So the goal is to enrich the spatial transcriptomic cell type tissue maps. So here is still the pipeline for this Visium technology. Each spot representing here is a circle, and actually each spot captures multiple mRNA, which is an mRNA mixture. And current deconvolution method estimate the proportion, but we actually still don't know what exact single cell expression in each location. So that's why we still need method to recover the single cell level expression. And after you know, let's say, how many cells in cell type A in location, then you can naturally calculate the proportion. So for this estimating cell type proportion, we can utilize the regression, dimension reduction, Bayesian, deep learning. Anything that can give you a weight type of the output, you can treat it as a proportion. However, for this inferring the single cell level data, it's actually we need to leverage the methods in the imputation or the mapping. So for the imputation, you want to impute the missing single cell information. And for the mapping, you want to transfer the single cell data to the spatial location, which exactly you want to map each single cell to each location. You can utilize the optimal transport. So given all of these categories of the methods, there are definitely benchmarking methods. So one representative benchmarking is the one published in Nature Communications. They benchmark about 18 methods over 50 data sets. And in terms of the performance, they list the card, cell to location, tangram are the best method for conducting the cellular deconvolution task. So the tangram is actually the method trying to mapping single cell to the spatial location. Also another method, they benchmark single cell and spatial transcriptomics integration for the distribution prediction and cell type deconvolution. And they also find that in these different methods, that the reference-based method also rank as the top method. So because the field evolves fast, so I leave some list some like open questions and hopefully can motivate some new ideas. So the first one is definitely there are advancements for the reference-free deconvolution. They need it for uncharacterized or even rare tissues which are lacking the single cell references, and also helps avoid annotation bias and review the novel cell type and also states. And the current challenge is determining the correct number of cell types. And it's also challenging to separate transcriptionally similar cell types, and also lack of consistent annotation pipelines. And also the robust deconvolution across multiple samples and multiple conditions. So all the methods that I introduced today only focus on one sample spatial transcriptomics deconvolution. However, now people are generating multiple samples, so it's essential for multi-sample, multi-condition, and atlas level analysis. And also we need spatially aware integration tools for comparative studies. And also, there will be method for performing multimodal deconvolution, combine the transcriptomics, spatial coordinates, imaging data, or even other omics modalities. And the challenges include constructing high-quality multimodal reference data sets and managing the modality-specific noise and the technical biases. And also, we need comprehensive benchmarking and validation because there are too many tools, but too few golden standard data sets. And most benchmarks limited to such as the brain tissue or low resolution data. And the future needs the benchmarks using diverse real-world data sets, evaluation of such as imaging-based and spatial transcriptomic integration effect, and also cross-validation frameworks with some pathologist input. So also now the field is moving to a very ultra resolution. So each location doesn't represent a mixture of the cell type, but instead represents a subcellular resolution. Can we utilize these high-resolution technologies and to do the deconvolution? So the new method need to aggregate these barcoded subcellular areas, better segmentation, and also label transfer and image integration. And deconvolution will remain relevant for both low-resolution and high-resolution data. So we need to reconstruct the single-cell transcriptomics from the spatially dense grids. So that's all for today. Thank you.